talking about. So somebody said to me that they preferred to know about the history rather than the biochemistry. But in discussing the history, I think we ought to ask, well, what's the use of it? And the use of it is that this 20th century and the 21st century are centuries of the gene. We have now come to a point where we understand that heredity is the transmission of genetic material from one generation to another. Okay? So you are here and alive because you're successful. And you're successful in terms of Darwinian principles because your parents had the right genes to be able to survive and pass on their genes to you. All right? So I'm going to go step by step. So I'm just going to do a quick revision of yesterday because we've got material to cover today. But stop me. Please stop me if I have to. So the unit of heredity is the gene, a sequence of DNA bases that codes for a specific protein. Now, yesterday I showed you this. I said it's a helical molecule and it's got only 4 bases or 4 letters: adenine, thymine, guanine, cytosine. And if you want to look at it more carefully, adenine always links with thymine and guanine always with cytosine. So it's a coil, it's a helical coil, and it is the unit of heredity. Now, I said to you that for your cell to be able to divide and make another cell, it has to pass on its DNA. Do you agree with that? And it only passes on its DNA if DNA makes a copy of itself. So the DNA has to first make a copy of itself, and the copy has to be absolutely precise, where these original sort of backbones have to be precisely copied, and there are enzymes involved in doing that. Okay? So when you make a second copy, it is exactly the same as the first copy. Right? Okay. Now, it all happens in the nucleus of the cell. So that DNA is strung in the nucleus on what you call chromosomes, but that's not an issue that we need to go into. Okay? Now, again I say to you that if you look at a DNA molecule like this, and you say, okay, this is DNA, right? And I say to you that this DNA, with this particular arrangement, with those 4 bases, adenine linked to thymine and guanine linked to cytosine, how does it code for the protein that is eventually going to make you? I think that's the question. And the way that happens is that one of the strands of DNA is usually copied to make a messenger, which you call messenger RNA. So remember, DNA stands for deoxyribose nucleic acid. RNA stands for ribonucleic acid. So this particular single strand is copied from here. So the information for making a protein lies in this double helix, and that information now is carried or passed on to the RNA. Is that okay? And this happens in the nucleus of the cell. Now, so if DNA is a string of billions of bases, these 4 bases, they are repeated billions of times and they lie in your nucleus, then what determines how different proteins are made? Because you have almost 100,000 proteins in your body. So the answer lies in the sequence of the bases in the gene. Now when I talk about the gene, so you got a whole lot of bases of DNA. That stretch of bases which codes for a specific protein is called a gene. So essentially you've got genes lying next to each other. But if you break it apart, you just see letters, adenine, thymine, cytosine, guanine. Is that okay? Okay. So now, the answer lies in the sequence of bases in a gene that code for a protein. Now look here, I've put these letters from the English alphabet down. O-L-S-A-H-I-T-P. It makes no sense. Does it make any sense? No. But look at the words you can make from it. You can make hat from it. You can make hit from it. You can make salt, pat, pit. And you can even make bigger words like hospital. Those make sense to you. So exactly like that, we've got 26 letters in the alphabet. We have 4 letters in DNA, right? But they can be in any sequence to form different proteins. So out of a particular sequence of 8 letters, we formed what is analogous to proteins, words, meaningful words. That's what DNA does. Out of its 4 bases and out of its particular sequences, it can make different proteins. How's that? Okay? Okay, right. Now, okay, so when that messenger RNA is made, okay, it leaves the nucleus. It leaves the nucleus and it goes into what you call the cytoplasm of the cell, and it sits on the structure here called an endoplasmic reticulum. And on that endoplasmic reticulum, there are these little beads called ribosomes. And it goes and sits plunk on the ribosome. It says, here I am, I'm a message from the nucleus, and I've brought it from my DNA. Okay, now, then comes along another kind of RNA. It's a cloverleaf structure, and it's called transfer RNA, right? Now, the transfer RNA says, okay, this messenger RNA has a sequence of bases here. It could be anything, adenine, cytosine, guanine, cytosine, adenine, thymine, right? Okay, so that transfer RNA says, I have got 3 bases here called an anticodon. I have to seek the complementary bases to what's on the messenger RNA. So if I had adenine here, then I must find uracil on the messenger RNA, because they complement one another. So we're going back to the original thing, where adenine always has to link with thymine, cytosine always with guanine, right? Now, in the RNA, it's not thymine, it's not this red one, it's uracil. But it doesn't matter. You can consider it to be the same. So the adenine will link to uracil. So now you've got these triplets here. 1, 2, 3, 1, 2, 3, 1, 2, 3, and they will find a complementary anticodon in this part of the transfer RNA, right? But this anticodon, say it's AUG, just say it's AUG, that means that it carries a specific amino acid, which is a building block of the protein. So the DNA sequence is going to determine the type of protein the cell is going to make. And there's so many different thousands of proteins, and there's only 20 of these building blocks or amino acids. So the sequence of the amino acids will determine the type of protein. Like in your alphabet, the sequence of letters determines the type of word you make. Here, the sequence of the bases determine the sequence of the amino acid, and the type of protein that is made is determined by the sequence of amino acids. Does that make sense to you? Right, okay. Now, so here we are. Here's the transfer RNA. It's carried an amino acid called methionine. It had on its anticodon UAC. So the adenine will always link with uracil. That adenine will link with uracil here, and the cytosine always with guanine. Do you see that? And so it will leave, it will actually have this amino acid. It says, okay, amino acid, you're going to stay here, and we're going to make a protein. And there's it. More of these transfer RNAs come along and they add to that one amino acid, and you get the growth of the protein chain. And now you're going to make a protein. So the whole story is here. You've got DNA that can make more DNA when cells divide. It can make a single-stranded RNA, a messenger RNA, which leaves the nucleus, goes and sits on the ribosome, and then determines from its sequence, which it got from the DNA, determines the type of transfer RNA that will come and attach to it, and that transfer RNA will have a specific amino acid attached to it, and the protein chain will begin to grow, and you will get proteins, and those proteins make you. So if you've got blue eyes, you'll say, oh, my father has blue eyes, my mother has brown eyes. I have inherited both genes, my father's gene for blue eyes and my mother's gene for brown eyes. But the protein that gave my blue eyes its color was dominant in my system, so I got blue eyes. It's as simple as that. Does it make sense? Oh God, I'm relieved. At least I'll have a sleep tonight. Okay, guys, we need to. So essentially, the whole story is DNA can make a copy of itself by replication. It can make an RNA by a single copy of a single chain. That's transcription, and that RNA gets translated into a protein. So this protein is determined by this sequence of bases, which come from the gene. So genes code for proteins. And that's the dogma of protein hypothesis. DNA makes RNA, which makes protein. DNA makes DNA, that's replication. DNA makes RNA, that's transcription. RNA codes for protein, that's translation. Okay, so let's go on to today's topic. Now, the question is, people always ask, so how much influence do genes have in shaping us into what we become? Is it genes only, or does the environment play the role? And it's the old nature-nurture debate. How much of what we are is nature, and how much is nurture from the environment? Now, we go back to this man who says we shouldn't look at our genes for a quick way to make life better. And he had this interview which I went through with you yesterday. But what I want to do for today's talk is I want to talk to you about epigenetics around 4 questions. All right? And our discussion will revolve around these following questions. First and foremost, we have all come from a single cell. A single cell, a fertilized cell or a fertilized ovum that contained material from your father's sperm and your mother's egg. You happy with that? Eventually, you've become you. Trillions and trillions of cells. Do you agree? So you can imagine how much the DNA had to replicate, how much RNA it had to make, and how each genes determine all the proteins that make you. Right? So this is beginning to make sense. So the fertilized egg has 23 pairs of chromosomes, one set from dad's sperm and another from mom's egg cell, giving us a total of 46 chromosomes containing all your genes, right? Now, so that egg, that single fertilized egg, starts multiplying. And it multiplies and multiplies and multiplies. And then it begins to differentiate, which I'll explain to you, and specialize. And you call that a division of labor, which is maintained for life. And that is what forms organs and organ systems in your body. So you see how important these proteins are. And these proteins were determined by DNA, and it's a very precise mechanism. It's got a lot of integrity because you're still alive. Gene expression in organs is specific both for protein coding and non-coding regulatory genes. That will come too, so don't worry about that. Now, the question is, what happens to the rest of the genome in the cell? Is it silenced or deleted or degenerate? Now I'm going to explain these things, right? So let's just go through the questions first. The second one is the twin question. Twins, both fraternal and especially monozygotic, which are identical twins, have more similarities than differences. Sometimes even monozygotic twins show differences despite being raised in the same environment or in the same home. Why? Second question. Third question. Many children who are physically and emotionally abused have serious problems later in life. But some who were abused in their childhoods grow up not to have these problems. Why? You read of this. Now, I don't have an answer to this question. This question is one that you answer by personal experience, where you read about or you know about somebody who was abused as a child, and the person either grows up to be an abuser herself or himself, or the person grows up to be an exemplary member of the community who actually goes out and forms organizations to fight child abuse. Right? So that can happen. And the fourth question is the question of transgenerational inheritance. What do we pass on as parents to our children and grandchildren? And there I'll discuss the Dutch hunger crisis. Okay? Now, let's go into this question. So, the answer to all the above questions is epigenetics. And epi means on or upon or over or beside. Now, it refers to all the cases where the genetic code alone is not enough to describe what is happening. There must be something else going on as well. Have we found the link between nature and nurture? Okay, so that's the question. Now, I've used this book called The Epigenetics Revolution. It was written by this woman, Nessa Carey, who is a British biologist working in the field of molecular biology and biotechnology. And she's written this book, she's written it wonderfully. It's actually brilliant. And it explains things very well. And then I also use Carl Zimmer's She Has a Mother's Laugh, Her Father's Brown Eyes, Heredity. We inherit traits from our parents through our DNA, our genes. And these traits are passed on to us by our parents through their genes. And the genes code for proteins, and you call that gene expression. So that's what I explained to you yesterday. Right. Now, let's go back in history. This is very interesting. There was this man, Jean-Baptiste Lamarck, who was born in 1744, and he died in 1829. Now, Jean-Baptiste Lamarck, he had a very long name. He was simply known as Lamarck. He was a French naturalist, a soldier, a biologist, and an academic. Now, he proposed the idea that biological evolution occurred and proceeded in accordance with natural laws. And he's right. That's how biological evolution takes place, according to natural laws. Life on Earth, he said, has evolved over long periods of time. Okay? Life on Earth has evolved over long periods of time, very slowly, little by little, and successively into a striking variety of organisms. Okay? Now, he's right there. Up to that point, he agrees with Charles Darwin. He's in agreement with Charles Darwin. But then he made another point, which took him completely off track. He said animals became more diverse as each creature strove towards its own optimal life function, hence the variety of living things on Earth. That was completely wrong. And I'm going to tell you why. Okay? He was wrong because Darwinian evolution is driven by random genetic variation combined with non-random natural selection, and some variations allow for adaptation and superior reproductive success over time. Now let's explain that statement. Now, what Darwin is saying is that from most of you here must have had children. From generation to generation, there is genetic variation. Okay? So your children will never have they will have your genes, but they're in a different combination. You must remember that you can't say, my child is a clone of me, because she also inherited half your partner's genes. So she would be different from you, although you her mother or her father. So from generation to generation, there is a variation. Okay? Now, that is a random process. And if you take any gene and you look at its history, that gene that you're carrying today, say for the expression of the protein albumin, if you follow the history of that gene, you'll find that the gene has changed over a period of time. Right? Changed how? A few changes in its bases. Okay? A few changes in its bases, as long as it's still a base that can find a complementary base to actually link with, right, in the DNA. So that is called a mutation. So if you are reading I'll tell you about it now. Okay. So Lamarckian evolution depends on the inheritance of acquired characteristics. Lamarck says your environment actually changes you. Now, he was laughed at then, but he wasn't completely wrong. But he made a ridiculous suggestion. He said, take this giraffe. It was originally a short-necked ancestor. But look at its progeny down the line of generations. It looked up at the tree, and then to reach higher, it sort of extended its neck. And then that one gave birth to this one, which carried on stretching, and that one gave birth to this one. And now they're very successful because they inherited this stretch. Okay? Now, stages in the evolution of the present-day giraffe, according to Lamarck. He said the short-necked giraffe, by constant stretching of its neck, would pass on a longer neck to its offspring. Now, that's like saying that if my father was circumcised, then I'd be born circumcised. It's not like that. It's not like that, okay? So environment does play a role, but not to the degree that Lamarck makes it so, all right? But he's not laughed at as much as he used to be laughed at. Okay, now, guys, this is just to show you that if you looked at your DNA, all you'll see is bases. That's all you'll see. Bases, right? And they're made up of 4 letters: adenine, guanine, thymine, and where's the cytosine? There would be cytosine somewhere here. There's a cytosine, right? So those are the 4 letters. Now, as I said to you, looking at this, there'll be a sequence. And this whole thing makes up one gene, all right? One gene. So it will express for a particular protein. And that protein could be trypsin, which is an enzyme, all right? But now, if you go back in time and look at trypsin genes in your ancestors, you will say, hey, you know what? My trypsin gene is slightly different from those of my ancestors. Instead of a C there, it had a G. Instead of a G there, it had a T. Because genes over time undergo random mutation. So they change, okay? And you call these single nucleotide polymorphisms, SNP. Now, let me tell you about mutations. We have them randomly, okay, over periods. Some of them are neutral. Can you hear me? Some of them are harmful. They could tell you that by age 45 you're going to get a certain cancer. And some of them are beneficial, right? You are a product of very beneficial mutations because you're here. Okay, so your mutations in your ancestors were good ones. So the genes they gave you are good and your proteins are good. Okay, now, so It's not so difficult, is it? Kath, is it difficult? It's not so difficult. Okay, so now, let's go back from a single cell. Now look at those mutations. Say what would happen now in a population of people. Can I give you an example? Say there were 1,000 mosquitoes here, right? And I was scared that I'm going to have a sleepless night because they're going to bite me. So I'm living in the modern world. I have mosquito repellents. I take Doom and I spray them on the 1,000. And say 995 of them die. Why didn't the other 5 die? Because they had a mutation in the genes which gave them a resistance to Doom. What are those 5 going to do? They're going to multiply and they're going to be successful in a new environment, in a doom-laden environment. That's what it is. That's how evolution works. So natural selection will then choose those 5 mosquitoes and they will go on to the next stage and be the new generation of mosquitoes. That's how it works. Random mutations can be beneficial, like they were beneficial to these 5 mosquitoes, but they were harmful to the other 995 because they died. They couldn't resist the gene. Okay, that's what it is. Now, let's go back. Now, guys, so you were once a single cell. Can you believe that? You were once a single cell. Now you are trillions and trillions of cells. Now, what happens in your instance is that all your cells can't be identical because then you'll be a useless blob. Okay? And what you need to do is you need to say, as my cell numbers increase, I have to actually compartmentalize cells, right? So what happens is you start differentiating cells as you grow in your womb, in your mother's womb. Right? You start differentiation. And you even gradually specialize, during which only a few genes express themselves in specific tissues while the rest are silent. Now I'm going to explain that, right? So, your father's sperm brought its 23 chromosomes. Your mother's egg cell had its 23 chromosomes. And then you had a fertilized egg, which is thought of as a single cell. And then that began to divide and proliferate. Okay. So the sperm and the egg have only 23 chromosomes. When they combine to form the fertilized egg, they reach the full complement of 23 pairs, which is 46 chromosomes. And chromosomes are the material the genes sit on. Okay? That's all you need to know. Now, look at it. Sperm meets egg, you had a fertilized cell, which then divided into 2. So yesterday's lecture comes in, it's DNA replicated, and so the DNA is identical now in these 2 cells, right? Then each of these cells go and divide and make 4. And then they start increasing in number. This is your path in life. This is how you came to be. So the fertilized egg was 1 cell, you got a 2-cell stage, you had the 4-cell stage, an 8-cell stage, a 16-cell stage, until the cells began to become too many, and so you formed a blastocyst, which has more than 1 layer of cells. Okay? So this was very early after conception. You're still a long way from being formed. So this is from a single fertilized cell. So this part here goes on to make all your organs. This part here makes the placenta. Okay? In your mother's womb. Now, so here it is. Just another diagram, just showing it to you in color of how the cells now, because they are too many to be just in a single layer, they're beginning to form multi-layers, and then they reach the level of the blastocyst. Now, look at this. This blastocyst is implanted within the first 2 weeks. Okay? It's implanted within the first 2 weeks. It has a couple of 100 cells. Okay? But, look at this. By 3 weeks in your mother's womb, some cells have become destined to be the cells of your brain and your nervous system. That is what I mean, that you have to have a division of labor. Some cells have to become specialized and become neurons to make the brain and the spinal cord. Are you with me? Then, by 4 weeks, besides the nervous system, you start getting cells that are going to become the upper limbs, and cells that are going to become the lower limbs, and cells that are going to become the skeleton, and then by 5 weeks, the eye, the ear, the lungs. By 8 weeks, you got the teeth, and as you go on and on and on, by 38 weeks, you are a fully fledged baby. You've got trillions of cells. Okay? And what made this? DNA expression made protein that actually formed you into this. Okay? And the way you're going to look from your phenotype, they say that she looks like her mother or she has her mother's laugh. Those were the genes that came from your parents, and those are the dominant genes that actually made you. Okay? So, there are all these sensitive stages in the development. We won't go into that. Now, this man here, Conrad Hal Waddington, throughout his career, he maintained that the arts were integral to science, and he continued to draw inspiration from the arts for his own work. So he tried to understand this process from one cell to millions of cells, together with the differentiation of cells, right, and their specialization. So many went into making the brain, many went into making the heart, many went into making the kidneys, many went into making parts of the gastrointestinal tract or your urinary system. So that is a division of labor. Okay? I always tell my first year class of medical students, all of you are first years, and you're all doing the same subjects. When I see you in 12 years' time, you weren't to be so homogeneous. You'll tell me you're a cardiologist, you're a urologist, you're a neuroscientist, you are a cardiothoracic surgeon, you have specialized. This is the specialization we're talking about here. So, here is the original cell. Now, this is Waddington's epigenetic landscape. That your mother's first fertilized ovum. It's totipotent. That means it can make any cell. It can make a kidney cell, it can make a brain cell, it can make a heart cell, it can make a skin cell. Okay? It's totipotent. It has in it the DNA information, right, to make any kind, to divide and make any kind of cell. Right? So you call it totipotency. The ability of a cell to give rise to all other cell types, body and placenta. Do you understand that? Okay. Now, as the cells begin to specialize, to divide and specialize, they slowly go down the slopes and they come to find their grooves and they sit there. What we're saying here is that white ball on top of the hill is the fertilized ovum. It rolls down to different troughs towards the bottom of the hill. You get different degrees of specialization. By the time you reach the bottom, you are highly specialized. Okay? So over here, this could become one of the skin cells, that could become one of the heart cells, that could be one of the brain cells, that could be one of the stomach cells. Okay, so now they're specialized. Now, the question is, once you're specialized, right, is it possible to go back and become totipotent again? Because once you're specialized, you already find yourself in the liver or in the stomach or in the kidney. But has that cell, can it go back and be a totipotent cell? So the question we're asking, and I think please listen to this and tell me whether you understand it. When you had a fertilized ovum, which was a single cell, and it was totipotent, and it could actually form, as it divided, it could form all these specialized cells that are different from each other. What we're saying is, you agree that the entire genome that you inherited from your mother and father was in that cell. Do you agree? Okay. Now, as it went down and specialized, okay, it is no use becoming a liver cell and producing protein that is important for brain function. Would you agree? It is no use being a heart muscle cell and producing pepsin, which is a digestive enzyme in the stomach. It doesn't make sense, right? So the question is, if you have in your genome, in every cell in your body, if you have 30,000 or 23,000 genes, right? And you become a liver cell, what are you going to make? In the liver, cells make albumin. So say in your liver cells make about 30 proteins, right? So you're going to express 30 genes. What happened to the other 23,000 and something genes? What happened to them? Do they just die off? Do they just die off? Do they become degenerate? That is the question. Because what we're saying is, if this is a liver cell, are you with me? And it's only expressing protein that is essential to liver function, okay? Can it go back and become totipotent and say, I am in a position to make any cell because my entire genome is ready for action? Whereas the liver cell will say, hey, Boot, I can only make cells that are important, I only make proteins that are important for liver function. Do you get the question? The question simply is, can you go back to totipotency? Right? And the question is answered by this man here with the long hair. His name is Sir John Gurdon, and his colleague Shinya Yamanaka. They showed that yes, it's possible to go uphill, and I want to tell you how they did that. Okay? So, they won the Nobel Prize for this work. He is an English developmental biologist at Cambridge. He is best known for his pioneering research in nuclear transplantation and cloning. He won a Lasker Award. He won the Nobel Prize in Physiology and Medicine in 2012 with Shinya Yamanaka. Yamanaka is the director for the Center for IP Cell Research and Application at Kyoto University. So they collaborated with each other and they found an answer to that question. Now, let me tell you how a scientist thinks. A scientist says, I have a hypothesis, okay, that as cells become more specialized, that means from your original cell, if you got liver cells, brain cells, heart cells, stomach cells, they've become specialized or differentiated, they undergo an irreversible repression of genetic material. That means that when the liver cell makes protein for the liver, all the other genes which are capable of making proteins for all the other organs, they are repressed. Okay, so what would be the possible outcomes when this hypothesis is put to a test? There's one possibility that the hypothesis is correct, and that once they differentiated, they can't go back. The adult nucleus, when inserted into an egg cell, will never be able to reproduce and produce tadpoles like the original fertilized egg cell, because by specializing, it has already lost or inactivated all its other genes. Or the hypothesis is wrong, and new toads can be created by removing the nucleus from an egg and replacing it with one from adult tissues. Okay, so here's the experiment. Now, when Gurdon said to his teacher in school, I want to be a scientist, the teacher wrote in his report, I believe Gurdon has ideas of becoming a scientist. In present showing, this is quite ridiculous, right? And Gurdon won the Nobel Prize. Okay, so what Gurdon did is he said, here's a frog, Xenopus levis, it's an African clog toad, right? Contains all the genetic material it had started with. So it started also, like you, as a fertilized ovum, right? Now, when it became, say, when some cells became skin cells, right? does that skin cell then inactivate some of the genes as the cell became more specialized? Do you understand the question? Okay. He took the nucleus of the cell from an adult toad, from the skin, right? And here is this nucleus. It's got the entire genome, but only certain genes are active because they are making proteins to keep the skin maintained, right? Okay. Then he took an egg cell, right? And he removed its entire genome and he put the skin cell genome into that. Okay? And he says, let's see if we can have a reversal to the original stage. So, this is the hypothesis, and this is just a revision. We said all cells become more specialized, they undergo an irreversible repression of some genetic material. What does this mean? As cells differentiate into specialized types, for example, liver, kidney, neurons, it is not uncommon for an organ to be made of different kinds of cells. For example, the stomach has oxyntic cells for acid secretion, chief cells for pepsin, G cells for gastrin. And then what is common in every cell in your body are the housekeeping genes, because they want to make ATP out of glucose to keep the cell alive. But what is different is that each cell is very specialized and makes only proteins for the organ it finds itself in. The rest of the genome is silenced. And in a diagrammatic form, so you had an egg cell which then went on and became differentiated, right? And now we're asking whether we can take a differentiated specialized cell from the frog skin and go back to the original state. He succeeded. He reprogrammed specialized cells. So the answer to his question is, whilst only certain genes were active in this organ, say the liver, the rest of the genes were silenced. They were not killed, or they were not thrown out of the cell. They were still there. Potentially, they could actually express any protein, but they were silenced because this was the liver, and for liver you only make liver protein. How's that? Got it? Now I want to tell you something. When you have a cancer, okay, you call this process regulation, right? So the genome is regulated in each cell in the body. When you have a cancer, right, you get deregulation. You suddenly see in your tumor, say you had a cancer of the stomach, right, and that stomach originally only made proteins for the stomach. And you have a tumor, and you look at the tumor cells in the stomach, they'll be expressing any protein. They become deregulated, and they run amok, and the cell becomes dysfunctional. It can't be a mucus-secreting cell anymore. It can't be a pepsin secretion anymore. It loses its specialization. It loses its differentiation state, and it becomes deregulated. So it produces any kind, right? So Gurdon says the potential is there, but they're silenced, right? And there was no detectable deletion or degeneration of any part of the genome in the specialized cell. The genome of which now had become totipotent. So, how? So if the rest of the genes in that specialized cell were not destroyed, how were they silenced? They were silenced by epigenetics, right? And here's a diagram, right? This is the genes. You know, like I showed you, say it's made up of 10,000 bases, right? Adenine, thymine, cytosine, guanine. And it's going to express a protein, like I showed you yesterday. Okay, but if you put in a chemical here called a methyl group, CH3, you can silence that gene. And that gene becomes suppressed and it doesn't express. If you don't put a methyl there, right, then that gene would express itself and make the protein. So you see, that is epigenetics, and this is the beauty of it. The beauty of it is that nothing has changed when it comes to the genetic story. The gene itself doesn't change at all. its regulation changes. It is told when to express and when not to express by a simple chemical that would come and bind to it nearby. Not even inside it, nearby it. You see, that's epigenetics. Okay? Now we're going to learn more. Now, you remember cytosine was one of the bases, right? How do you add a methyl group to a gene? When you add a methyl group, you just take your CH3, which is a chemical, and it adds to the cytosine, and the gene stops expressing. So all those silent genes in your specialized cells are methylated. The ones that are expressing are unmethylated. Isn't that easy? Okay. Then there is another kind of gene, another kind of regulation. Now, let me tell you about these genes. What I didn't tell you about them is they are long strings of these bases, right? But if you lay them out, they'll cover a lot of space. But your cell is something so small, you can't even see it with the naked eye. And packed inside its nucleus are 23,000 genes. And you can't have them spread out. So they are like knitting wool around a golf ball. And the golf ball in this case is a histone or a protein. So they packaged, okay? And these histones are proteins, and the genes wrap around them. The DNA wraps around them, okay? Now, if you add an acetyl group, can you see AC here? It's referring to this acetyl group. AC, if it binds to histones, you turn the gene on. Remember I said when you methylate, you turn the gene off. When you add an acetyl group, you turn the gene on. And so, here it is. By gene methylation and gene acetylation, you are silencing or activating genes to function. So there's an example. That's your chromosome. On it are a whole lot of genes, DNA, which coil around these proteins called histones. If you want to silence a gene, you put in a methyl group on the cytosine, like I just showed you. And if you want to activate a gene, you just add an acetyl group to a histone. So, central dogma still stands. DNA makes RNA, makes protein. Okay. There is another thing. Look here. I told you yesterday that when you've got DNA and it contains all the information for making a particular protein, it first has to hand over that information in the form of bases to an RNA, right? And that RNA then went on to become a protein. Now look here. Here's the DNA, right? Remember, it can replicate and make a copy of itself. It can transcribe into RNA, and the RNA can translate into protein. Would you agree with me? Right. But sometimes the DNA makes an RNA which is a non-coding RNA. That RNA can go back to the gene. It doesn't go to the protein stage. It goes back to the gene and silences it. So that's another way of silencing. These are ways that our cells, our specialized cells, in our brain and our heart and our livers, behave. They silence when you need to silence, they activate when you need to activate. Nature is so precise and so regulated, right, that if you didn't have these regulatory functions, you'll be growing teeth in your eyes. That's what the book says, right? Or you might have some organ which is not supposed to be on your nose, growing on your nose. That could be pretty embarrassing for you. Okay? So this is such a beautifully regulation method. Now, guys, let's go to the twin question, right? We're running out of time. Don't leave me just yet. Like you did last night. Twins, both fraternal and especially monozygotic or identical twins, have more similarities than differences. Would you agree? Sometimes even monozygotic twins show differences despite being raised in the same environment. So identical twins show differences. Why? Now, Steven Pinker says that identical or monozygotic twins are far more similar than fraternal twins, whether raised apart or together. That's an important point. Biological siblings, whether raised together or apart, are far more similar than adoptive siblings. But if the DNA sequence was all that mattered, identical twins would always be absolutely identical in every way. But they're not. I've read papers where they have identical twins, one schizophrenic and the other is not. Why? And they have the same genome, identical. So there is something that is non-genomic that's acting on the genome and causing these differences. Okay, so epigenetics can drive a wedge between twins. Identical twins are monozygotic. One develops a disease and the other. You call this discordance. Did the one with schizophrenia spontaneously develop mutations in her or his brain cells? Or were there environmental differences? It is hard to say because the early environment of these twins, identical twins, is quite the same. You never know what the subtle differences in environmental differences, even for monozygotic twins, are anyway. An extreme scenario is one dies early because he's run over by a bus. But that's a different story. Now, epigenetics in schizophrenia. If one pair of an identical twin is affected, there is a 50% chance of the other having the condition. But why is it not 100%? Because identical twins have the same genomes. Thus, individuals may be genomically identical, but have different outcomes in life. Okay? Now, the study was done by Manuel Estella, and he said that the chromosome, the chromatin material in infant monozygotic twins, right? If they have a little difference in methylation, like I showed you now, adding a methyl group, or histone acetylation between the two twins, what happens is, by the time they reach their 50s, they've got more differences, especially those identical twins who have lived apart. Because they are in a different environment, they have different methylation and acetylation of their genes. This is consistent with the idea that the epigenomic differences are the result of environmental differences. That means that twins have identical genomes, but environment plays a role in forming the final product. Okay? So Jeffrey Craig in Melbourne, he did the same experiments, right? He did DNA methylation in identical and fraternal twin pairs. Fraternal twins had more differences than the monozygotic twins at birth in specific parts of the genome, right? But in monozygotic twins had some differences at birth, right? Changes take place in utero in fraternal and monozygotic twins. Conclusion, even twins diverge epigenetically in utero, and the differences become more pronounced in adulthood. So we are not only a product, my original question, we are not only a product of our genes, we're also a product of environment interacting with genes. Okay? Differences in outcome of monozygotic twins can also be explained in principle by identical predisposition, which evokes different outcomes depending on different environmental challenges. And then there's the other challenge. Say one of the twins smokes, the other doesn't, but only the smoker gets lung cancer. So that's an environmental factor, right? The demonstrated epigenetic differences shown by Estella and Craig may just be correlations and not causal. That's important. Now, abusive childhoods. Many children who are physically and emotionally abused have serious problems throughout their lives, but some who were abused in their childhoods grow up not to have these problems. Why? I haven't got any stuff on it, but a child less than 3 years of age is abused and neglected by her parents. She is given for adoption by the state. She is then raised in a home with love and affection. Some do well, and others have a substantially higher risk of adult mental health problems than the general population, but can have no recollection whatsoever of their early trauma. And then I want to go on to the last question, transgenerational inheritance. from parent to child to grandchild. So when we have these changes in a particular environment, do we pass those